Hello and welcome to today's lesson. In previous lessons we've looked at enzymes and how they work, we've looked at the digestive system, and today we're going to look at Required Practical 5, where you are expected to investigate the effect of different pHs on amylase activity. So by the end of this lesson you should be able to recall how to carry out an investigation into the effect of different pHs on the activity of amylase. You should be able to explain why starch and iodine are used to investigate the effect of different pH on amylase activity, and you should be able to interpret the results of an experiment to determine the rate at which amylase is working. So in previous lessons we've spoken about enzymes, okay? And an enzyme, just to refresh your memory, that's E-N-Z-Y-M-E. That's like the smallest bottom bit to a Z ever. An enzyme is defined as a biological catalyst. Now what that means in plain English is that they speed up chemical reactions without being used up themselves, and they do that within living organisms. You've got enzymes in your body that are currently doing the exact thing I've just described. Now enzymes work on what are known as substrates. So a substrate is a substance that fits into the active site of an enzyme. Now if you're watching this video and you're thinking, I don't know what Mr. Abraham's going on about, then you need to go back and watch the video on enzymes, please, because you're going to struggle today, if not. Now, amylase is an enzyme that breaks down starch. Okay? So amylase is an enzyme that has an active site. So if this is my amylase enzyme, so this is my amylase. The substrate that amylase breaks down is starch. So that's my starch, which is the substrate. Now what happens is that starch will go into the active site of amylase and it will be broken down into products. Okay? Now exactly like we've said earlier, if you're looking at this and you're saying, I don't understand that, you need to go away and watch the video on enzymes, please. So amylase is an enzyme that breaks down starch. Now we've also said in previous videos that all enzymes have optimum conditions. So the optimum conditions can be defined as the conditions at which an enzyme works best. At which an enzyme works best. Now, conditions that can be at their optimum for enzymes include temperature and pH. So these include temperature and pH. So amylase has a temperature at which it works best, and it also has a pH at which it works best. Now, another substance that you've heard about in previous lessons is starch. And starch has a particular test for it, which you need to be reminded of to understand this practical. So can we put the subheading, please? Test for starch. Now, we looked at the test for starch when we did the food tests practical in practical 4. Now, I've got some diagrams ready here. Essentially, when we test for starch, we can take a boiling tube. So this is my boiling tube. Boiling tube. We can take a pipette and we can add iodine to it to see if starch is present. So we can add iodine to see if starch is present. See if starch is present. Okay. If we add iodine to a solution, iodine is normally orange by itself. So iodine will stay orange if there is no starch present. However, if starch is present, iodine will turn blue or black. So we can put this diagram down here. I can make it a bit smaller so I can fit some writing in. And we can say that if starch is not present, iodine stays orange. So if starch is not present, iodine stays orange. But if starch is present, iodine turns black. So if starch is present, comma, iodine turns black. Okay? So if starch is present, iodine will turn black. So the important thing you need to know from this is that iodine remains orange if starch is not present, but turns black if starch is present. Okay, so you need to know that we add iodine to starch to see if it is present or not. Now, we're going to do an experiment to find the best pH at which amylase works. So we can say that we want to investigate the optimum pH for amylase. Okay? So our independent variable, the one thing we are going to change, so the independent variable, which is the one thing we're going to change, is the pH. So we're going to be changing the pH of the solution. And we can do that with what's known as a buffer. A buffer is a substance that keeps the pH the same. So a buffer, B-U-F-F-E-R, solution keeps pH at the same value. It doesn't let it change, okay? Our dependent variable is going to be the time taken for the starch, or for the, iodine to stop turning black. Okay? Our control variables, which are the things we keep the same, control variables are going to be the temperature, the volume of enzyme, so the volume of, not enzyme, we'll put amylase, the volume of amylase, starch, or iodine. And you could also say the concentration of amylase, starch, and iodine. However, I haven't got room, I don't think. I'll try. And the concentration of amylase, starch, iodine. So we've said so far that enzymes are biological catalysts. They break down what are known as substrates. The substrate fits into the active site of the enzyme, and the optimum conditions are the conditions at which the enzyme works best. We've said the test for starch is that you add iodine to it. If it goes black, starch is present. If it remains orange, it is not present. We are going to investigate the optimum pH for amylase. Therefore, our independent variable is going to be the different pHs. We can control that using a buffer solution. A buffer solution keeps pH at the same value. Our dependent variable is the time taken for iodine to stop turning black. And the control variables are going to be the temperature, the volume of amylase, starch, and iodine, and the concentration of amylase, starch, and iodine. So let's think about how we are going to investigate this. Okay? Please remember this is a pre-recording, so if you don't get the notes down in time, you can press pause. So we need now a method. And I'm going to put it with a different color actually. So method. To carry out this investigation, the first thing we are going to do, so number one, is we are going to place a drop of iodine into the wells on a spotting tile. Into the wells on a spotting tile. Okay, now a spotting tile looks like this. Okay, so a spotting tile is like a plastic square with these little wells inside of them. So into each of these wells, we're going to put some iodine. So this is my spotting tile. And I'm going to use a pipette to put a drop of iodine into each well. So I'm going to put my pipette there, and I'm going to right next to it add a few drops of iodine to each well. So once I've added my drops of iodine, remember iodine is an orange colour by itself. So I've added my iodine to each well in the spotting tile. Okay, I've only added a couple of drops. I don't need to go overkill. I literally need to use 2, maybe 3 drops at most. So I've added my iodine to the spotting tile. Now, what I'm then going to do is I'm going to take, so number 2, I'm going to take a boiling tube and add 1 centimetre cubed of amylase and 1 centimetre cubed of buffer solution. So add 1 centimetre cubed, so 1 cm cubed of amylase and 1 centimetre cubed of buffer solution to 5 test tubes. Okay, to 5 test tubes. So I'm going to take my boiling tubes, which look like this. I'm going to take 5 of them. So 1, make it a bit smaller. 1, I've done that wrong, because if I take 1, 2, this is really annoying me now because they're going to be slightly bigger and smaller. 3, 4, and 5, and into each test tube or boiling tube I'm going to add 1 centimetre of amylase and 1 centimetre of buffer solution. I'm going to do that using pipettes. Okay, so I'm going to take some pipettes and I'm going to add my amylase and my buffer solution. So I'm going to add amylase with 1. So I'm adding 1 centimetre cubed of amylase, and with this one I'm going to add 1 centimetre cubed of buffer solution. So into each one I'm going to add 1 centimetre cubed of amylase and 1 centimetre cubed of buffer. Now remember, the buffer will keep the test tube at the same pH. So if you use buffer solution 5, it will keep it at a pH of 5. If you use buffer solution 7, it will keep it at buffer solution 7. Okay? Now you're going to take your test tubes and you're going to add them to a water bath. So number 3, take the 5 test tubes and add them to a water bath set at 37 degrees Celsius. So you're going to set the water bath at 37 degrees Celsius and you're going to put your test tubes in them. Now a water bath looks like this, and it's used to keep things at a set temperature. So you're going to take your boiling tubes containing the amylase and the buffer solution and you're going to put them into the water bath. You're also going to add your beaker of starch solution to the water bath. So also add the beaker of starch solution to the water bath. Okay. Once you've added your test tubes and your starch solution, you're going to leave them in the water bath for 5 minutes. So leave these in the water bath for 5 minutes. Okay, so you're going to leave them in the water bath for 5 minutes. Once you've left them in the water bath for 5 minutes, you're going to take one of the test tubes and you're going to add starch solution to it. Okay, so number 4, after 5 minutes, add the starch solution to one of the test tubes but keep them in the water bath. Okay, so you're going to keep them in the water bath, but you're going to add the starch to one of the test tubes. You're then going to immediately take a pipette and add 1 drop of the contents of that test tube to a well in the spotting tile every 30 seconds. Now this is hard to visualize. I'm going to show you how to do that in a moment with pictures, okay? So, number 5, take 1 drop of the contents of the test tube take 1 drop of the contents of the test tube each or every 30 seconds and add it to a well in the spotting tile. Okay, add it to a well in the spotting tile. You do this until the well inside the spotting tile no longer turns black. So we can just put next to this. Continue to do this until the iodine in the well no longer turns black. You would then repeat this with the other test tubes and then repeat the whole thing with a different buffer solution. So number 6, repeat steps 4 to 5 for other test tubes, and number 7, repeat steps 1 through to 6 with a different buffer. So you would get a spotting tile and you would add drops of iodine into each spotting tile. You would then take 5 test tubes and into each test tube you would add 1 centimeter cubed of amylase and 1 centimeter cubed of buffer. You would then take your 5 test tubes and put them in a water bath for 5 minutes set at 37 degrees Celsius. You would also put in there your conical flask containing starch. They have not been added together yet. After 5 minutes, you would take some starch solution with a pipette and add it to the test tube that's been in the water bath. You would then start taking a single drop of the contents and adding them to each well every 30 seconds and recording the colour change. Okay? If you haven't got this method written down, write it down now, because on the next slide, I'm going to clear the screen. On the next slide, I'm going to explain this last bit because it's hard to visualize. So, what you've got is your spotting tile, into which you have added your iodine from earlier. Okay, I've done that wrong. I'm just going to leave it like that. So this is your spotting tile containing iodine. So that's your iodine, like so. So we can put underneath here, spotting tile containing iodine. Now I'm going to remind you of what we said on the very first slide in red, that iodine will turn black if starch is present. Okay, so iodine will turn black if starch is present. I'm going to remind you as well that amylase is an enzyme that breaks down starch. So we can also say amylase is an enzyme that breaks down starch. So what we've done on the previous slide is we've got a water bath. So we've got our water bath set to 37 degrees. In fact, I'm going to put it a bit closer. So this is our water bath. Set to 37 degrees Celsius. Inside that water bath, we've got some test tubes. Some of the test tubes contain buffer and amylase. So this is our test tube with buffer and amylase, and we've also got other test tubes in there containing starch. Now these have been left in the water bath for 5 minutes, okay? So I'm going to label that one test tube with starch. Now, what you are asked to do is to use a pipette to add some of the contents of the starch into the test tube containing amylase. So I'm going to use a pipette to remove some of this solution and then add it to this solution. So I'm going to use a pipette to add starch to amylase slash buffer. Once I've done that, every 30 seconds, I'm going to use a different pipette to add some of the contents of this one into one of these wells. So I'm going to use this pipette to take up the amylase and starch, and I'm going to then add it to this one after 30 seconds. I'm then going to take some more and I'm going to add it to this one after 60 seconds. And then I'm going to take some of this again and add it to this one after 90 seconds. Okay? So I'm going to add amylase and starch every 30 seconds. So if I'm adding it every 30 seconds, what I might find is that my results will look something like this. So this is going to be my spotting tile that I'm looking down at. This is going to be my 30 seconds, 60, 90, 120, 150, 180, 210, and so on and so forth. When I add my first drop to this 30-second tile, it will go black. And what that means is that starch is still present. Okay? So if it goes black, starch is present. If it remains orange, it is not. So black, starch is present. Whereas at orange, starch is not present. Okay? So every 30 seconds, I'm adding my dots at 30 seconds, 60, 90, and then I might find that at 120, it's gone orange. So it's gone from having starch present to the starch not present. Therefore, at 120 seconds, the starch has been broken down by amylase. So at 120 seconds, the starch is no longer present as it has been broken down by amylase. So the amylase has broken down the starch at 120 seconds. Now let's assume that this is at buffer 4. If I was to do this at, say, buffer 9, which is much more alkaline than buffer 4. So if this is at buffer 9, I may have added my amylase to the starch at this one, 60, 90, 210, sorry, 260, 30, 60, 90, 220, sorry, 120, 150, 180, 210, and then it may have gone orange down here. So this time we can say that at, so this 30, 60, 90, 120, 150, 180, 210, 240, 270, 300. So at 300 seconds, starch is no longer present as it has been broken down by amylase. If you look at these two, the amylase work much better at pH 4 than it did at pH 9. Therefore, we would say that amylase works better in acidic concentrations because it broke down the starch much more quickly here than it did in this one. So from these results, amylase works best at pH 4. Okay? So the final thing we need to do is we need to think about how we could evaluate some results. So we need to draw a results table. I can't do that with a pencil and ruler purely because I'm doing it on a computer. In your books, I would like you, please, to do it with a pencil and ruler, as it is academic policy and it is, better practice. So let's have a think about how we can evaluate some results. So I need a white pen. We're going to have down the first column, time in seconds. So time in seconds. We're going to have another line. In fact, we'll just put this in, save changing the pen again in a minute. 30, 60, 90, 120, 150, 180, 210, 210, 240, 270, 300. Along the top, we're going to have our colour of iodine solution. And underneath there, we're going to have all of our different pHs. So we're going to have pH 2, pH 4, pH 6, pH 8, pH 10. Okay, these are the different ones we're going to have. And I'm going to cordon those off. And rather than writing the different colours each time, I'm just going to use a black. Oh no, I'm going to have to use a blue and an orange pen, okay? So let's say for pH 2 that at 30 seconds it was still black, 60 black, 90 black, 120 black, 150 black, 180 black, all the way down it was black. That would mean that the enzyme did not break down the amylase. The amylase did not break down the starch, okay? So amylase did not break down starch. Now what we would find with your results is that you would get the same over here. At pH 10, you would probably get the same results. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 2, 3, 4, 5, 6, 7, 8, 9, 10. You would get exactly the same results. However, Around these areas, you would start to find that you would get some starch being broken down, okay? But what you would find is that at one of the pHs, off the top of my head, I can't remember which one it may be, but one of the pHs would break down the starch much quicker. So you would find that, for example, pH 6 broke down the starch in 120 seconds, whereas pH 4 broke it down in 240. Once it went sort of a yellowy colour, usually you would stop, okay? Just because once it's broken it down, you don't need to continue. So you would say, you would conclude from this, that amylase works best at pH 6 because it broke down the starch the quickest. So amylase works best at pH 6 as it broke down the starch quickest. Now, if we wanted to work out what's known as the rate of these reactions, so rate is how fast something happens. Rate, how fast a reaction happens. We can do this by doing 1,000 divided by the time. So rate is equal to 1,000 divided by the time. So we can work out our rate at the different ones. So for pH 4, we can do 1,000 divided by 240. So if I can just get my calculator, 1,000 divided by 240 would give me a rate of 4.16, and we would give that the unit of speed over 1. So because this broke it down at 240, I would therefore say that it is 1,000 divided by 240. For pH 6, I could do 1,000 divided by 120 to give me a rate. So 1,000 divided by 120 to give me a rate of 8.3. Unit of speed per time. For pH 8, I could do 270, or 1,000 divided by 270 to give me, I can work it out, 3.7 seconds per unit of time. So if we look at these results from this results table, by doing this calculation of rate, we can find that at pH 6, the rate at which the enzyme amylase worked was 8.3, whereas at pH 4 it was 4.16, and at pH 8 it was 3.7. Therefore, it works best, or it works the fastest, at pH 6. This has been a very long lesson. It's a very difficult practical to understand, so I apologise that I've rabbled on for so long. Hopefully by now you can recall how to carry out an investigation into the effect of pH on enzyme activity. You can explain why starch and iodine are used to investigate the effect of different pH on amylase activity, and you can interpret some results to determine which amylase is working at the best rate. If you have any questions about this lesson, please let me know via email or Google Classrooms. If not, I will see you in the next one. Thank you very much.